Psychedelics. They're good for more than having a deep conversation with a houseplant. These drugs have huge potential for treating a variety of mental illnesses, too. Studies show that people with depression, PTSD, and substance abuse disorders sometimes experience fewer symptoms after a dose of LSD, for example. There's just one problem. We don't really know why. And since the side effects of these drugs aren't exactly subtle, it would be great if we could find a way to develop new medications with all of the antidepressant power and less of the acid trip. Luckily, a researcher named Bryan Roth at the University of North Carolina has been studying this stuff his entire career. His research, mapping the fundamental chemistry of the brain and the ways these drugs work, has provided some clues as to how psychedelics can improve symptoms. Roth and his team have even synthesized new molecules that could have the same effect without the hallucinations, which sounds like the kind of research we would like to see in person. We sent our friend Madelyn Leembruggen to Roth's lab to learn more about this far-out solution to some of psychiatry's most stubborn problems. This is SciShow Field Trips.
Thanks, Hank. I'm here visiting Bryan Roth's lab at the pharmacology department at UNC Chapel Hill. Pharmacology is all about drugs, but for Roth, it isn't just the drugs that are fascinating. It's what they interact with, chemical receptors.
In Montana, I went to this really small college, Carroll College. By then I was convinced, I needed to study the brain. I need to study these drugs and how they work, basically. But I had no concept on how to do that. Mm-hmm. One year, this scientist came. Ostensibly, he was there to give us a lecture. But I think he was really there to go trout fishing, basically. I think that was the reason he was there. Uh-huh. But he gave his lecture. His lecture was on how the brain works. And he mentioned that, neurotransmitters work through receptors. And he said, this is where drugs act, are these receptors. So I said right there, okay, so I need to study receptors. So that's it, basically. Mm-hmm. I saw the word receptor. I said, that's what I want to study. That's where drugs act. And that was it.
And I have to say, I can see why he was so excited about it, because the way this works is really cool.
This is where we sectioned a brain with an optical microscope. And this is the distribution of the receptor here in various brain regions. That's sort of a close-up. So these are these neurons here in the cortex. These are called layer 5A neurons.
This is a picture of neurons?
Yeah, neurons that are expressing the receptor.
All the chemical receptors in our bodies have a unique shape, and when a certain molecule comes along that fits into those receptors, they can activate. Scientists call these activating chemicals agonists. Neurotransmitters are a type of agonist, since they're the signaling molecules that neurons use to send messages throughout the nervous system. Now, even though textbook diagrams of receptors are just still images, when you zoom down to the cellular level, every molecule in our bodies is constantly in motion.
The way people usually describe receptors and drugs is that the drug is the key and the receptor is a lock, okay? That's actually not the way things work at all, okay? Not even close. So technically speaking, that's called the induced fit model. Our understanding now is what's called conformational selection. So the idea is that the receptors are vibrating. They're continually vibrating, and they will vibrate in a conformation that the drug is specific for. And that binding event causes a small change in the overall equilibrium of the population. It's like the locks are transiently opening, and the key is just popping in.
Basically, the agonists that can only fit a little bit of themselves into the receptor aren't going to send a very strong signal, and the ones that bind to more surface area will. It's way more complicated than that, trust me, which is why people like Roth have made a career out of studying these little things. As a graduate student, Roth focused on opioid receptors. But when he got a job at the National Institutes of Health, he switched gears and started studying something called 5-HT2 receptors. 5-HT is an abbreviation for the molecule 5-hydroxytryptamine, which is the neurotransmitter that binds to these receptors. But you're probably more familiar with its common name, Serotonin. We have different kinds of 5-HT2 receptors all over our bodies because serotonin does a lot more than just regulate your mood. Serotonin plays a role in everything from digestion to your immune system and even your blood clotting. And while serotonin and 5-HT2 receptors are buddies, it isn't the only molecule that can bind to them. Another molecule that likes to get groovy with 5-HT2 receptors is the drug lysergic acid diethylamide, or LSD.
So LSD is a psychedelic drug, and there are a number of psychedelics. The drug mescaline, which is a psychedelic drug from cactus, peyote cactus, psilocybin for magic mushrooms. So all of these psychedelic drugs have the same property that they activate this receptor, the 5-HT2A receptor. And activation of this receptor is responsible for their psychedelic effects in humans.
LSD is a semi-synthetic compound that, you've probably heard of before. It partially comes from a compound found in fungus that we've refined into something with more of a kick. Well, not me personally, other people. When LSD binds to 5-HT2 receptors, it activates the same pathways that serotonin does. And it also affects our emotional and information processing to the point where people experience hallucinations and disconnect from reality.
So these layer 5 neurons, these are very important for integrating information from basically all areas of the brain, all sensory areas and perceptual and cognitive areas. And what psychedelics do when they activate, when they hit the receptor on those neurons, is they basically cause the neurons to fire sort of in this disorganized fashion. And then, for reasons that are not entirely clear yet, the mind starts paying attention to sort of randomly generated information, sensory information, that it then makes a story about, okay? And that's the psychedelic experience. Basically, the part of the brain that tells us what reality looks like, it's basically like you injected noise into that system. And so the awareness, our mind, doesn't trust that information anymore.
Since its most popular use has been recreational, LSD is classified as a Schedule I controlled substance by the United States government. That's the group of drugs that are said to have a high risk of dependency and no established medical benefit. But how do you find out if there's a medical benefit if it's very difficult to test the stuff? That's been the issue.
Right now, it's like a really hot field. So I don't know how long that's going to last, but right now it is. So it'd be great if there was money. So it's not like there's been any more money that's been appropriated to study psychedelics. So there are all these people who want to study psychedelics, but there's no money to study psychedelics.
The limiting factor in the reaction.
Money is always a problem, yes.
Since LSD binds to the same receptors that serotonin does, there's reason to think it could have some similarities with antidepressant medications. The most common type of antidepressants are called SSRIs, or selective serotonin reuptake inhibitors. And what they do is right there in the name. After a neuron makes and releases molecules into a synapse, those molecules pretty quickly get broken down or sucked back inside the cell. That's called reuptake. SSRIs work by preventing serotonin reuptake, usually by stopping the proteins that physically transport serotonin.
Long term, probably what they do is they cause compensatory interactions in the circuitry, and that causes the antidepressant effect. So it's a very indirect effect, usually takes 6 to 8 weeks for the maximum response, and it doesn't work for everybody.
So current antidepressants sort of indirectly juice up the signaling that's already happening in our brains, rather than directly binding to and activating 5-HT2 receptors. But SSRIs don't work well for everyone, especially people with really severe cases of depression, and we don't entirely understand why. Like Hank said, there's some clinical evidence that taking LSD can also improve mood for people with certain mental health conditions. And with LSD, patients sometimes reported feeling better for months after a single dose.
Let's say you're depressed, and I started you on Prozac, I would say, probably 4 to 6 weeks before you're going to have much of an effect. With psychedelics, 1 dose, basically. Many people, at least according to the early trials, were no longer depressed after a single dose of psilocybin. And 12 months later, they were fine.
But we're still a long way off from having any LSD-based options at the pharmacy. I mean, there's that whole makes you hallucinate and distorts your reality thing. Not everyone's down for that as a medication side effect.
I presented that idea at the neuroscience meeting, I think in 2017 or something like that. I gave one of these presidential lectures. There were like 10,000 people in the audience. It was this humongous auditorium. I, presented that data, and on the slide, one of the slides, I had a bullet point. It simply said that it may be possible to separate the therapeutic effect from the psychedelic effect. And then about a year later, I got a phone call from DARPA, the people that invented the internet, GPS, stealth bombing, all these great things. So there was somebody from DARPA that was at my talk, because it turns out that in the military, the number one casualty is psychiatric. So by far and away, they have more soldiers who are disabled because of depression, anxiety, post-traumatic stress disorder, and so on, way more than physical casualties. And, they had seen the promise of psychedelics, but there's no way you're going to give psychedelics to somebody with a gun. It's such a bad idea for so many reasons, basically. So they contacted me and they said, is it possible, basically? I sort of hedged my bets and I said, well, it's a hypothesis we could test.
So Roth is trying to figure out how LSD might produce those effects and how to create similar compounds. And he's starting with the fundamentals. He wants to know what molecules like LSD look like when they're bound to a 5-HT2 receptor, as in what shape do they take and how strong is the signal that gets produced from their bond. Like any lab working with a controlled substance, there are precautions in place to make sure the research is safe and focused.
Yes, this is the safe.
Okay.
That is the infamous LSD safe.
Not quite what you were expecting, is it?
So we have LSD and many, many other Schedule I psychedelic drugs. The safe is kept in a locked room. That's one of the things. So the room we came in is locked. The other thing is that there's only one person in the lab that has the combination to the safe. And that is not me. We don't want anyone accidentally ingesting these compounds. We have psychedelics that are more potent than anything that is out there. But the amounts that we have, if we were to open that up, it's just dust, basically. Just little tiny amounts.
They only need an amount of LSD smaller than a microdose, called a nanodose, since all they need to see is one molecule binding to one receptor.
So when we initially solved the structure of LSD with the serotonin receptor, one of the things that we noticed was that there was a conformational change when LSD bound to the receptor, so that a lid came over the top of the receptor and occluded it. So it stopped LSD from exiting the receptor. So LSD was sort of trapped in there. Concomitant with this, there was a different type of signaling event that occurred, which is technically called arrestin signaling. And the normal mode of signaling through the receptor is what's called G protein signaling. Okay.
Basically, both G proteins and arrestins are molecules inside your cells that help pass along messages after a neurotransmitter binds to a receptor. But depending on which messenger takes over, it can have very different effects. When Roth's team realized that LSD seemed to push the signaling toward the arrestin pathway, they saw an opportunity.
So we had the idea, well, maybe, if we make a drug that's targeting the other pathway, the G protein pathway, maybe we can diminish the psychedelic effect and enhance the antidepressant effect.
All of this progress would have been just a pipe dream for researchers like Roth, even just a decade or two ago.
For like 20 years or so, my goal was to, solve this structure of the receptor. And we couldn't do it, basically, because there wasn't appropriate technology. And it wasn't until, probably 10 years ago that you could crystallize membrane proteins and study them by X-ray crystallography. And my lab did that. We actually got the first structure of LSD with the serotonin receptor by crystallography. But it was extraordinarily difficult.
Today, structural modeling is much easier. Roth's lab now uses a tool called cryogenic electron microscopy, or cryo-EM for short.
So I am a structural biologist, So what I do is I purify receptors, either from overexpression systems or from tissue, and subject those purified receptors to structural approaches like cryo-electron microscopy.
That's Nicholas Wright. He's a postdoc working to analyze the molecular structure of these candidate compounds.
So the kind of idea behind cryo-EM is that the purified material is put on these grids, and you form really thin vitreous ice, so glass-like ice, by plunge freezing the sample in liquid ethane. And the cooling happens so fast, the ice doesn't have time to form regular crystals. And what that does is it allows you to get images of single molecules of your receptors, and then with computational approaches, you can average all those together to get a crisp, clean, three-dimensional picture of your protein at the kind of really high resolution. So you can actually see the drug and how it interacts with the receptor.
So because it's frozen so quickly, then you can use image processing to take out all of the ice molecules, all the water molecules, and just focus on the candidate structure.
Exactly. Yeah.
Okay.
And because it's vitreous or glass-like ice, that gives very low background in the images. And you kind of capture like a snapshot of those molecules in action, and you can compute a three-dimensional high-resolution picture of them.
They're currently trying to discover new molecules that could also bind to the 5-HT2 receptor and exploring what effects those molecules might have once they get there. This kind of molecular matching game also used to be a really slow and difficult process.
So this is our robot that is preparing compounds that we're testing. And down there you can see it's taking little aliquots from the drug plate. All those plates down there are the drug plates. And then it's going to add them to assay plates. You know, this is repetitive work.
Right.
So it's perfect for a robot to do.
But thanks to the latest generation of software, researchers can now estimate what drug binding might look like for billions of slightly different molecules that no chemist has even synthesized yet. One promising category of molecules is the tetrahydropyridines, or THPs.
We had hooked up with this amazing chemist at Yale, Jonathan Ellman, who had invented this new chemistry to make this class of compounds called tetrahydropyridines. He had figured out a way to make, in theory, billions of tetrahydropyridines. You know, we can't test a billion compounds physically, right? But he could enumerate them computationally. So using his reactions, we were able to enumerate, I think, 100 million tetrahydropyridines that in theory he could make, but no one has ever made before.
They calculated the odds of them binding with what's called docking scores. The better the score, the more likely it is that researchers will try to actually synthesize it in the lab to test it out.
So the way we do drug discovery is first we have a structure. The structures are generally solved in my lab. And then collaborate with this amazing computational person, Brian Roth at UCSF. And his docking program basically takes the structure of each little molecule and then brings it into the receptor and then sort of tries to find a way in the receptor that it will fit. And then it scores that. You know, if it forms a hydrogen bond, it gets a score, ionic bond, et cetera. And currently, I think we're running a docking campaign with 4 trillion molecules. And then, you know, the compounds literally have never been made before. They don't exist in the physical universe. So then we have a chemical company just make them, and then we test them. And then they go here. This is the testing being done right here.
When they find that a compound binds to a receptor in the test tubes, they then move on to testing it in mice. And in this case, they found something really exciting. 2 of these molecules worked like strong antidepressants, similar to what some people report after taking LSD. But these new molecules didn't have the psychedelic side effects that LSD does, at least as best as they could tell. Measuring the psychological state of animals is notoriously difficult and has to be surmised from things like head twitching, which mice do when hallucinating. Even so, this could be the first step towards a whole new class of antidepressant medication. And other potentially powerful molecules are even further along in the process, but not for illnesses like depression. When studying another kind of 5-HT2 receptor that is involved in appetite suppression, Roth and his collaborators identified a molecule they called BMB-101.
So BMB-101, this is a compound that was discovered in a collaboration with Alan Kozakowski, who was formerly at University of Illinois, Chicago, and Bill Wetzel, who's at Duke. So we first published on this in 2015. We were developing these as potential antipsychotic drugs. They weren't very effective antipsychotic drugs. Alan, by accident, found in a zebrafish screen. Zebrafish are these model organisms. So they had a zebrafish screen which looked for drugs that are effective in treating seizures or epilepsy. And it turned out that BMB 101, also known as lumocastrin, was very effective in that screen. And it just finished phase 2 clinical trials for seizures. I think caused a more than 70% decrease in the seizure incident and also improved REM sleep, which is like unheard of among anti-seizure agents. So most people with seizure disorders actually have impaired sleep. So this, could be a game changer.
Roth thinks researching other compounds that bind to different 5-HT2 receptors could produce new medicines for a huge number of disorders, especially those that are treatment resistant. And for Roth, that really hits home, because one of the people who could have been helped by his research was his mother.
Yeah, so my interest in pharmacology actually started at a very young age. My mom was diagnosed with schizophrenia when I was 5 or 6. Of course, that had a huge impact on me. And she was sort of in and out of it was in Montana. And it was basically the state mental institution. It was really pretty awful place. So she was sort of in and out of that when I was growing up. And I think when I was 13 or so, my sister ended up in the front page of the newspaper for having taken LSD. And I looked up LSD, and it said, LSD causes a model psychosis. Okay. So I thought, okay, so this is what I need to study. I basically knew what I wanted to do, but I had absolutely no concept of how to get there. And it was just this chance interaction with visiting with a trout fisherman, probably.
That's what led him to pursue biology and chemistry in college, and is what brought him to that fateful lecture with the image of a receptor, and all the decades of studying them since.
We were ultimately able to get my mom on one of the newer atypical antipsychotic drugs, this drug called quetiapine. Transformed her life. And its action is to block that receptor. At the time, there was really nothing known about this receptor, okay? We knew that this drug clozapine, which was an antipsychotic drug, bound to it, but it wasn't clear that had anything to do with its actions. And it wasn't until actually a few years later that Richard Glennon discovered that it's the receptor for LSD. And the thing I was studying is sort of this pharmacological curiosity. So it was a complete accident of fate.
Roth's work has shed light on how some of the drugs for disorders like schizophrenia work, and how other medicines could be made with fewer side effects. He essentially helped create a field of science that many thought was impossible when he was starting his career.
I applied to medical school, and I applied to some MD, PhD programs because I wanted to be a scientist. One of the places that interviewed me was Johns Hopkins. I went there, and I got to my last interview, and he said, if you could study anything in the world, what would you study? And I said, I would study the chemistry of consciousness. And he said, you cannot study the chemistry of consciousness. He said, it is impossible to study the chemistry of consciousness. And in fact, that's what I do. And that's what I've done for the last 30 years.
SciShow field trips are made with our friends at HHMI Tangled Bank Studios. We've come together to bring you face-to-face with researchers at the cutting edge of scientific discovery. You can watch more of Tangled Bank science content at tangledbankstudios.org.